Hello, I'm Javairia Haq and I did prompt 2B. The genetic code is a set of rules used by living cells to translate information encoded within genetic material into proteins. But how is it discovered? Marshall Nierenberg and Johann Mattei both wanted to discover how information gets transmitted from DNA to proteins. In 1961, Nierenberg and Mattei set out to do an experiment to find the answer. They created a stable system by rupturing E. coli bacteria cells, releasing the cell's contents. The remaining cytoplasm could synthesize protein when RNA was added, allowing the researchers to control the experiments. Nierenberg and Mattei used 20 test tubes, one for each amino acid. Each test tube contained the cell-free E. coli extract, the poly-U homopolymer, which was synthetic RNA made up of only uracil units, and 20 amino acids, with one of them being radioactively labeled and the other 19 unlabeled. This allowed them to determine which amino acid would be incorporated into a protein. The tubes were incubated at 37 degrees Celsius and translation took place. The proteins had precipitated to the bottom and the free amino acids were on top. The protein was then filtered and the filter was checked for radioactivity. The results showed that the tube in which the protein was radioactively labeled contained newly synthesized protein with the amino acids specified by the homopolymer. In this case, the poly-U strand codes for phenylalanine. They then repeated this with other homopolymers and found that the poly-C strand coded for proline and poly-A coded for lysine. The poly-G homopolymer did not work. Their conclusion was that RNA controlled the production of specific types of protein. But what about the RNA strands that weren't just homopolymers? That's what Lieder and Nierenberg were out to discover in 1964. They used very short artificial RNA sequences called trinucleotides, as well as ribosomes and 20 charged tRNAs with amino acids, with one being radioactively labeled and the other 19 unlabeled. The ribosome binded to the mRNA as well as the tRNAs that were specified by the mRNA. The mixture was then put through a nitrocellulose filter which allowed for unbound tRNAs to pass through, but did not allow the ribosomes with the unbound trinucleotide to pass through. The sample was then tested for radioactivity. Nierenberg and Lieder found that if the filter paper was radioactive, then the trinucleotide coded for the radioactive amino acid. Through this experiment, they were able to identify 61 out of the 64 codons and organize them into a coding dictionary. And here are the sources I used.